This is an experimental setup that many teachers will recognise. It consists of a beaker of sodium hydrogen carbonate solution, and under this glass funnel there is an aquatic plant. And as the plant photosynthesises, it releases bubbles of gas which are being collected and they're displacing the sodium hydrogen carbonate solution. Traditionally, the organism that was used was an aquatic plant called Elodea, which is readily available. Many people have it in ponds in their gardens, or you can get it from aquaria shop. But the problem with Elodea is that it is very, very temperamental. Sometimes it will respond and you'll get a sprig of Elodea that'll bubble very enthusiastically. Other times the rate of bubbling is very, very slow. So this means that within your class you can have two sets of students whose experience of the practical is very different. This plant is called Cabomba. The fantastic thing about Cabomba is it always bubbles. You can see here that the bubbles are streaming off. And its response to the light intensity is almost immediate. For the students to see that a plant is responding to its environment so rapidly is something that perhaps for many of them they've never thought about before. Using this apparatus, you can put it onto a sunny window ledge, leave it there for several days, and hopefully by the time you come back there'll be enough gas collected so that you could remove the test tube and use it to relight a glowing splint, which would demonstrate to the students that the gas you'd collected contained oxygen gas. This apparatus is fantastic for a demonstration, but for students to carry out individual investigations, all they'd need would be to take a boiling tube with some sodium hydrogen carbonate in and a sprig of the Cabomba to carry out a whole host of investigations into factors affecting the rate of photosynthesis. You can vary the distance of the light source from the tube, so that would allow you to investigate the effect of light intensity. With students lower down the school, they can quite simply count the number of bubbles that are produced at each different distance. With more senior students, you could collect the gas, which would then mean that you have quantitative data which you could use to plot graphs, carry out statistics. So you can tailor the investigation to the level of your students. Another interesting thing that you can do with your aquatic plant is you can wrap a filter, a light filter around it, and by using different coloured filters you can investigate the response of the plant to the wavelength of light that it's being exposed to. With a red filter, you'll find that you get a far greater rate than if you repeat the experiment with a green filter. And in this way, the students can create for themselves their own absorption spectrum, which will further their understanding of why it is that photosynthesis is not necessarily a very efficient process, because only certain wavelengths of light can actually be used by the plants to carry it out. Cabomba can be bought from aquaria, so it's available pretty much all around the year, but you can keep it in your prep room in a fairly deep tank in a solution of water, or add some hydrogen carbonate to it. In my experience, it does prolong the life of the Cabomba if you keep a sprig of Canadian pondweed, Elodea, in with it as well. So that will just mean it'll maintain your population for a little bit longer. It is a tropical plant. It's meant for use in fish aquaria, so it's very important that when you've finished using your Cabomba that you dispose of it in with household waste, and that you don't attempt to throw it into ponds or rivers, because it's not a native species. Cabomba really is wonderful, because when you plan to do this practical, you know that you're going to get a practical that works, that gets plenty of data that the students can analyse, and that they're really going to understand the process that you're talking to them about. With Elodea, you could never guarantee that. So I think the use of Cabomba in investigations for photosynthesis in aquatic plants really has revolutionised this investigation.